In this lecture, I'm going to describe skin. It's often referred to as the integumentary system. The integument is a system of the body because it contains all the basic tissues, and that's why we call it a system. We'll see later on during this lecture that skin contains epithelium and glands derived from epithelium, the exocrine components. It contains connective tissue, particularly the dermis of skin. It also contains muscle and nerves. So that's why we call it a system. Now, during this lecture, I would like you to assemble information that will allow you to then answer all the questions that I list here in the learning outcomes. I'm not going to go through them all here, but I will go through each of these in the course of this lecture. Skin has got many, many functions. And during this lecture, I'm going to explain the structural components of skin that enable these functions to be fulfilled. First of all, let's make sure we can understand the layers of skin. In this section, on the right-hand side, there is an image of a section through thick skin. There are two main different types of skin, thick skin and thin skin. And really, the term is a little bit confusing. Here is an image of thick skin because the epidermis, a layer I'm going to describe in a moment, is very, very thick compared to thin skin, where it's much, much thinner. Here is the epidermis. It's this surface layer. It's an epithelium. It's a very special epithelium. It's a stratified squamous epithelium because there are many, many cell layers making up the epithelium, as we'll see in a moment. But the layer on top, that pale purple layer, thick layer, happens to be keratin. And that's a very important component of skin that I'll describe later on. But this thick skin is found on the palms of our hands or the soles of our feet, where we have a lot of wear and tear. But really, as I said before, the term is a little bit confusing because really the thickest part of our skin is on our back because of the thick underlying layer, the dermis, indicated here. The thinnest part of our skin on our body is the eyelid or perhaps the scrotum. In both those areas, the eyelid and the back, the epidermis is probably the same thickness. So, as I said, the term is a little bit confusing. During this lecture, when I refer to thick skin, I'm talking about skin on the palms of our hands and the soles of our feet. On other occasions, I will refer to thin skin. But it's easy to describe the structure of skin looking, first of all, at this piece of thick skin. You know, sometimes we call skin either hairy or non-hairy. That's another way in which we can classify skin. And it's probably an easier one to understand. We certainly don't have hair on the palms of our hands or the soles of our feet. So the two main layers indicated here are the epidermis, the epithelium, it's derived from ectoderm, and the underlying dermis, which is derived from mesoderm. The dermis is dense connective tissue. When you look at it, it's very pink stained because it's full of connective tissue fibres. And that dermis supports the epithelium, makes our skin very strong. Underneath the skin, underneath the dermis, you can see a very clear layer. That's the hypodermis. It contains adipose tissue or lots and lots of fat cells. And if you look very, very carefully in that hypodermis, you can make out some little round circular profiles, stained bluish. These are sections through sweat glands, one of the skin appendages I'll talk about later on. They're all derived from the epidermis. And during development, they grow down and become very specialised in areas of the dermis, and as you see here, in areas of the hypodermis. Well, let's concentrate first of all on the epidermis. And as you see listed on the left-hand side of this slide, there are 5 main layers. And I'm going to go through these layers in some detail now. First of all, let's look at the stratum basale. It's often called the stratum germinativum because it's a germinating layer. It's a single layer of cells, a single layer of epithelial cells, tightly bound to the underlying connective tissue, the underlying dermis, by very, very large numbers of hemidesmosomes. These are macular adherens, special bonding structures that bind epithelial cells to the underlying connective tissue. Well, the nuclei of this stratum basale lie very close together, and that's because there's not a lot of cytoplasm associated with these cells. These cells divide constantly because skin is continually renewed. They divide constantly, and as they divide, they move towards the surface to form the other layers of skin. The stratum spinosum is a thicker layer. It consists of several layers of cells. It's often called the prickle cell layer or the spinous cell layer. And that's because if you look very, very carefully in this section, better still if you had the opportunity to look at this layer under higher magnification, you'll see between the cells there's a fine little clear space. And if you look carefully into that clear space, you'll see these little tiny spike-looking processes. These represent an artifact. In actual fact, in reality, the cells of this layer, the stratum spinosum, are linked together by long cell processes like my fingers here interlocking. And the fingers, my fingers here, are joined to each other by very strong desmosomes. So skin, the cell layer, the stratum spinosum, the cell processes are joined together from one cell to another by very strong desmosomes joining the process together. Like desmosomes are holding my fingers together, it's very hard for me to separate my fingers apart because of those junctions. That makes this layer of skin a very strong layer. It maintains the strength of the epidermis. Now, during processing, tissue shrinks. So the cells shrink apart and create spaces between the cells, and you see these projections or processes like my fingers are showing here. They're the spinous processes of the cells. They're the prickles, and that's why it's called the prickle cell layer. These cells then move up, and as they move up, they accumulate little granules of keratohyalin. This is the very beginnings of these cells transforming into being squames of keratin. These keratohyalin granules accumulate in the cells. They're very, very strongly basophilic, dark blue staining. The stratum lucidum is the layer that really is initiated as being the strata that the cells begin to die off once they've accumulated all this keratin. You only see it in thick skin, and it's not described that often as being a major component of the skin epidermis. It's often a very thin, very basophilic line that you see here. The top layer, the stratum corneum, is the layer you see at the surface. This layer consists merely of squames or cells that have died off and accumulated an enormous amount of keratin. The cells, or these squames of keratin, are lost from the surface of skin as a result of wear and tear, but as they move up from the stratum granulosum into the stratum corneum, the desmosomes I mentioned earlier that hold the cells together disappear. They're broken down. And that's how the squames or the keratinized components are released into the exterior. Now these squames, or these dead cells full of keratin, also have, during their final development, a very thick cell layer, cell membrane layer. And on the inside of the cell layer of this membrane is a insoluble protein. And on the outside of the cell membrane is an insoluble lipid layer. And these two layers, the protein and the lipid layer and the keratin, are our waterproofing component of skin. Sometimes lipid-soluble components can move through the epidermis. And that's used often in therapeutics. For instance, the nicotine patch that people sometimes wear to try and get rid of their addiction to smoking and nicotine allows the nicotine to be lipid soluble and move through the surface of the epidermis into the underlying tissue and help treat that condition. So the stratum corneum then is a very important layer. It's our waterproofing layer in skin. And as I mentioned earlier, it's very, very thick in thick skin. Well, let's look at the dermis now. Let's first of all have a look at the way in which the epidermis attaches to the underlying dermis. The interface between the epidermis and the dermis is corrugated, it's uneven. And that's because projections of the dermis, called dermal papilla, push up into the overlying epidermis. And also epidermal components bury down, invaginate into the dermis. And that creates a large surface area of the interface between the epidermis and the dermis. And because it's such a large surface area, then the epidermis is tightly bound to the underlying dermis. Now, sometimes, if you see in this image, you can see the surface of the keratin layer. It's sort of undulating, goes up and down. These actually reflect, to some degree, the random and different arrangements of these dermal papilla and the epidermal pegs in the thick skin. And those attachments, or those ways of increasing the surface area of the attachment, the dermal papilla and the epidermal pegs, are reflected on the surface as our fingerprints. And our fingerprints are very different between individuals. It's a reflection of the different random array of the arrangement of these dermal papilla and epidermal pegs. The dermis has got two layers, and they're best seen when we look at the dermis very close to the junction between the epidermis and the underlying dermis. It has a papillary layer, which is a very thin layer, just immediately adjacent to the epidermis. It's loose connective tissue, consisting of only type 1 and type 3 collagen, the lesser, stronger collagens in connective tissue. Underlying that is the reticular layer, and you can see it's a lot denser. The collagen fibres there are arranged very differently. They're thick collagen fibres. They make up that very thick reticular layer of the dermis and give strength to the dermis and therefore strength to the epithelium. Immediately below the dermis is the hypodermis. It's not really termed or classified as being part of skin, but all skin layers have, to some extent, underlying fatty tissue called the hypodermis. It's really a layer of adipose tissue of varying thicknesses. It helps us be insulated. It stores energy. It stops us from losing body heat. It's a very important layer. Sometimes in gross anatomy we refer to it as subcutaneous fascia. But again, it's just adipose tissue, fat cells. Here you see it here, clear staining, because during processing, the components, the lipid droplets, the fatty droplets are lost from the cell during normal histological processing. Sometimes in the skin, there are muscle attachments. In the platysma, which is in the neck region, and in the face, the muscles of facial expression and also the platysma are striated muscle. And they insert into the dermal areas of skin, into the strong dermis of skin you see in this image here, rather than inserting onto bone. The erector pili muscle, which is really the muscle that I've listed on this slide, is a very important muscle, particularly in animals. It attaches the root of the hair follicle to the very strong dermis. And in animals, when that erector pili muscle contracts, it can make the hair protrude a lot taller and make the animal look a lot scarier. It's not so important in humans. But in humans, sometimes that erector pili muscle contracts and creates what we experience as being goosebumps. Let's look now in more detail at the cells that make up the epidermis. We've already looked at the different layers of the epidermis, which reflect the process of keratinization and our waterproofing role of skin. But let's look at what cells are present and what they do. First of all, we've actually mentioned the keratinocyte before. It's the cell, as you see here, stained with a special stain that shows you really the accumulation of keratin within the cell. It's predominantly the cell type in the epidermis. It is the important cell layer that is in contact with our external environment. And as I've told you in a previous component of this lecture, its main job is to produce the waterproofing agent keratin and those insoluble proteins and lipids on the surface layer of the stratum corneum. Let's look at the melanocyte. The melanocyte is a cell in the epidermis that produces the pigment melanin. And melanin is very important because it protects the layers of the stratum basale, the cells in the stratum basale, the nuclei in the cells, and also the nuclei in the layer above it, the stratum spinosum. It protects those nuclei from excessive ultraviolet radiation. And the process of creating melanin is a very, very rather long process, a rather complicated physiological process, so I'm not going to describe it here. But what I want to emphasize is that when the melanin is produced by the melanocyte, it's donated to the surrounding keratinocytes. The surrounding keratinocytes actually ingest the melanin pigment by endocytosis. And then the melanin is transported and surrounds the nucleus. Here's an image of skin. It's actually thin skin. So have a look now and recall the section of skin that I showed earlier. The layers are much thinner. The keratin layer is much thinner. So this really does describe the difference between thick skin and thin skin that I mentioned before, purely being really the difference in the thickness of the epidermis. But what I want you to concentrate on here is that dark brown pigment you see in the basal layers of the epidermis of this thin skin. That's melanin. And if you look very, very carefully, you'll notice that the bulk of the melanin is concentrated above the nucleus, between the exterior surface and the nucleus, again shielding those nuclei from excessive ultraviolet radiation. Those nuclei, remember, are in cells that are dividing constantly. So you don't want that excessive ultraviolet radiation damaging the DNA during that division process, during the process of mitosis. Let's now move on to the Langerhans cell. It's originated from bone marrow. It moves into skin from bone marrow. It's a dendritic cell. And what I mean by a dendritic cell is that it has long processes. You see these processes in both these images. They're antigen-presenting cells, and they live mostly in the stratum spinosum. And have a look at the image on the right, the one with a pink background to it. Have a look carefully into that pink background. You can just make out the profiles of circular nuclei. They belong to the nuclei of the keratinocytes of the stratum spinosum. These cells, as you can see here, not only have long processes, but they also hold hands with each other. These processes are joined together from one cell to another. They link together. Now, I use the analogy that it's like chicken wire. It's like the wire of a fence. And that's what these Langerhans cells or dendritic cells actually create in the epidermis. A whole layer, if you like, of chicken-like wire cell structure. And if you're an invading pathogen trying to get through the epidermis to underlying connective tissue and therefore the rest of the body, it's almost impossible to pass through that epidermis and not come into contact with one of these cell processes of these dendritic cells, these antigen-presenting cells. And when a foreign component, a bacteria or an antigen, comes in contact with these cell processes, the antigen-presenting cell then ingests that antigen, processes it, and then puts it on the surface of the cell. And that cell then leaves the epidermis. The antigen-presenting cell leaves the epidermis and travels to a nearby lymph node. And there it puts a little flag up on the surface and alerts T cells and other cells of the immune system that then recognise this antigen and mount an immune response against it to help protect our body. And we'll learn about that in more detail in a later lecture. Another cell type is the Merkel cell. It's also a dendritic cell, meaning it has long processes extending through other cells, through other layers of the epidermis. These cells are hard to see. They're very difficult to see, and they tend to be situated at the very base of the epidermis. They're mechanoreceptors. Well, skin is also a very large sensory receptor organ. In fact, skin is the largest organ in the body. It occupies between 15 and 20% of the volume of our body. So it's a huge area, and it's very, very important that the exterior coat of our body has a number of sensory components in it, sensory receptors, that detect certain sensations, like pressure, like temperature, those sorts of sensations. But I just want to introduce you to a couple of receptors that you can actually see when you look at sections of skin. First of all, there are a number of receptors that have free nerve endings. These are simply nerve endings ending within the tissue that detect certain sensations. And they're hard to see. You'll never see these looking through sections of skin using normal routine stains. So I'm not going to introduce those at this stage. What I'm going to emphasise are two encapsulated nerve endings. By encapsulating, I mean that they're enclosed by certain connective tissue components or capsules. And I'm referring mainly to the Pacinian corpuscle and Meissner's corpuscle, because Ruffini's corpuscles, again, are structures that are very hard to see using routine histological processing. Here are two images of Pacinian corpuscles. They're stained differently, but they show the same histological features. They're mechanical receptors. They detect pressure and vibration. And if you look at any one of these two Pacinian corpuscles, you can see that they have a very tiny small dot in the centre. That's the axon, the neuron. And then that axon, that neuron cell process, is surrounded by numbers and numbers and numbers of sheets called lamellae. You can see them clearer on the right-hand image. Those lamellae are really wrapped up encircling Schwann cells. And on the outside is a thicker stained capsule. It looks a bit like a section through a cut onion. Now, fluid lies between all those lamellae. And when pressure or vibration is applied to the surface of the skin, then that distorts the capsule and it distorts these lamellae of Schwann cells. And that distortion is then transmitted through the fluid to then initiate a stimulus on the axon, and therefore the information is conveyed back to the central nervous system for interpretation. Let's see now how we can identify a Meissner's corpuscle. These are touch receptors, and you find them concentrated in places on the body like the lip or the fingertips or the toes, places where it's really important to have a very precise sensation of touch. They're a bit harder to see, but they lay up in the dermal papillae, those projections of the dermis up into the underlying areas of the epidermal pegs, or up into the superficial layer of the actual junction between the epidermis and the dermis. On the left-hand image, you can see these two projections of the dermal papillae into the epidermis. And look very, very carefully, particularly in the more central projection. And you can make out a fine, light pink stained structure with some bluish stained nuclei. That represents coils of Schwann cells. And those coils of Schwann cells wrap around an axon. You can't see the axon in that H&E section, but have a look across to the right-hand image. Stained differently, you can just see a very fine, thin black line towards the base of the image. That's the axon, stained with a special stain. And that axon is going to project all the way up and be encapsulated by these Schwann cells. That's typical of a Meissner's corpuscle. Again, as I said, it's a touch receptor. Well, Ruffini's corpuscles are mechanoreceptors, and as I said, they're very difficult to see, but they respond to stretch or torque of the collagen fibres within the dermis, and therefore give us some information about the sorts of shear forces or stress forces on our skin. Well, Let's now look at skin appendages. Again, they're epidermal in origin. During development, the epidermis burrows down into the underlying dermis and becomes specialised into various structures. Shown here in this diagram is a picture of the three-dimensional structure of skin, and it shows you on the right-hand side diagram the epidermis and hair projecting from the surface. Underneath there are various other structures that are derived from the epidermis. Sweat glands of two types, eccrine and apocrine, are also derived from the epidermis. They're an example of these epidermal appendages. We'll talk about these in more detail in a moment. Hair follicles, that are areas where the hair is produced and renewed. And associated with hair follicles are sebaceous glands. They're all skin appendages that have very important roles in the function of skin. Let's look at the sweat glands in more detail. There are two types. Eccrine sweat glands that are widely distributed over the body, and also the apocrine sweat glands that are restricted mainly to the locations of the body I've listed there, particularly the axilla. Here's an eccrine sweat gland. It consists of a secretory portion that I've labelled here, a paler stained portion of the gland, and a duct portion, which is a darker stained area. Much smaller profile the duct portion has than the large, wider lumened secretory portion. If you look very carefully at the secretory portion, it's also surrounded by myoepithelial cells, which are contractile cells, and they help to dispel the sweat. On the right-hand side image, you can see, if you look towards the left-hand edge of this image, the duct proceeding its way through the dermis and up through the epidermis. Now, the best way to imagine what a sweat gland looks like, I think, is to think of a cooked piece of spaghetti. Next time you go home and cook spaghetti, get a piece of the spaghetti and hold it up in your hand and stand next to the edge of your bench, your kitchen bench, and lower that piece of spaghetti until it comes in contact with the bench, and then keep lowering it some more. What happens is that the spaghetti will start to coil up. What you hold in your hand is the duct portion, and what's coiled down the bottom on your kitchen bench is the secretory portion. So when you cut through that piece of spaghetti at various levels, you're going to see different profiles of the spaghetti, such as what you see here in this image. You can see parts of the duct system cut transversely, perhaps sometimes longitudinally or obliquely, the same with the secretory portion. Now, when we secrete sweat, we're doing two things. We're secreting water, which evaporates from the surface of our skin. So sweat has a thermoregulatory role. It cools the body. The evaporation of that water or sweat from the surface cools the body. So one important function of sweat is thermoregulation. Other components of sweat are components of the body that we want to get rid of, such as uric acid, urea, ammonia. And therefore, sweat glands are also not just a thermoregulatory organ, but an excretory organ. Here's an apocrine sweat gland. It's got a wider lumen, a larger luminal space lined by cuboidal-like epithelial cells. There are also myoepithelial cells wrapping around these secretory components that help to eliminate the secretion product. The ducts are usually the same size as what you see in a sweat gland, But remember, they do not modify the secretion product. Eccrine sweat glands, the ducts do modify the product. These apocrine sweat glands become active during puberty, and they secrete proteins and pheromones. And these are odourless, but they can be acted upon by bacteria and create an acrid odour. It's important to understand something about the innervation of these sweat glands. They're both innervated by sympathetic divisions of the autonomic nervous system. But in both situations, the neurotransmitter substance used is different. Eccrine sweat glands are cholinergic. They're said to be cholinergic, whereas apocrine sweat glands are said to be controlled by adrenergic nerve fibres. And I'll explain that in a lot more detail when I lecture on the nervous tissues. But that brings about different reactions, because when we secrete sweat with the role of thermoregulation, we first of all sweat from our forehead, and then our scalp, and then the rest of our body, and finally the palms of our hands and the soles of our feet. But when we sweat because we're emotionally activated or we're stressed, we start secreting sweat from both the eccrine sweat glands and also the apocrine sweat glands, particularly the axilla. There, we tend to sweat first on the palms of our hands and the soles of our feet. So the different stimuli for sweating, whether it be by the eccrine sweat glands or by the apocrine sweat glands, are controlled by separate types of fibres from the sympathetic nervous system, cholinergic and also adrenergic. Let's briefly look at the structure of hair and a hair follicle. It's not something that we emphasise a lot in histology of the skin, but shown here are three profiles through sections of the hair at different levels as they travel up through the dermis before the hair finally protrudes from the surface of skin. On the left-hand side, you can see the dermal papilla projecting into the base of the hair follicle. That dermal papilla is no different to the dermal papilla projecting up into the overlying layer of epidermis that I described before. The very surface of that dermal papillae has on it epidermal cells, typical of the epidermis, stratum basale, and also melanocytes that are going to contribute to the colour of hair. And then on the outside of the hair follicle and on the outside of the hair as it proceeds to the surface are a number of root sheaths, the internal and the outer root sheath. The outer root sheath is really just a representation of the downgrowth of epidermis. And the internal root sheath is really a few layers of soft keratin as it's produced and gives rise to the more firmer shaft of keratin in the centre of the hair. And on the remaining two slides, the one in the middle and the one on the outside right-hand side, you see a transverse section taken through the hair, showing different layers of the hair. On the far right-hand side, you see a central white unstained region which represents the hair shaft, and two layers on the outside that represent the hair cortical and the hair cuticle regions of the hair. But again, as I said earlier, we don't emphasise really a lot about the histological structure of hair in our lecture on skin. Associated with each of these hair follicles, though, are sebaceous glands that I'd like to describe here. Sebaceous glands secrete sebum or oil. It keeps our hair oily and it keeps the surface of our skin oily. And again, these sebaceous glands are formed by downgrowth of epidermis, by downgrowth of the external root sheath that I mentioned earlier. And sebaceous glands are an example of holocrine secretion. Holocrine secretion is where the cell accumulates its secretory product and then the cell dies, it disintegrates by a process called programmed cell death or apoptosis. And here, in this image, you can see the very basal parts of the sebaceous gland, the cells being derived from basal stem cells. They accumulate their secretory product, the sebum, and then finally towards the luminal space associated with the hair follicle, the cells die and all the secretory contents, the sebum, is liberated into the luminal space. Let's finally look briefly at the nail. On the right-hand side, you see a section through the tip of the finger. You can see typical epidermis on the top left-hand part of this image. You can see a yellow stained component. That's a section through the nail, which is just keratin. Again, formation of nail is just like the formation of keratin on the surface of our skin, and also like the formation of the hair. But there's 4 or 5 major components of this fingernail, or this section through the fingernail, that I want to point out to you. First of all, the nail plate is a part of the skin where the epidermis loses its layer of keratin as it comes around to start to form where the nail is going to exit. And then along the nail bed, all you have really is remaining components of the epidermis, the stratum basale and the stratum spinosum, because you're not producing typical keratin that you do on the surface of skin, you're producing the nail. And underneath the nail is the nail root. That's the area that germinates the keratin, the layers of keratin, germinates the cells that are going to go through the process of keratinization, forming the nail root and the nail matrix. And as more and more keratin is formed, the fingernail is going to start to proceed and protrude from the surface of the finger. There are 2 parts of the fingernail that are very, very important. That is the eponychium and the hyponychium. And those 2 components, the eponychium and the hyponychium, are areas that seal the fingernail, the underneath surface of the fingernail, from the exterior. The eponychium is the cuticle region of the fingernail. It stops pathogens from getting into underlying germinal layers of the fingernail. And the hyponychium is where the nail protrudes from the finger, and immediately underneath that nail, again, is a region that you don't want pathogens to be able to access the germinating layers of the nail bed and the nail root. Well, I hope you've learnt now a fair bit about the structure of skin. Make sure you understand the layers of the epidermis and all their functional significances. Make sure you know the difference between thick and thin skin, and know all the functions of skin, particularly the functions that are served by all the cells that make up the epidermis, and the function of all the skin appendages, and also know how to recognise those cells and also those skin appendages. So thank you very much for listening to this lecture. Again, I hope you've enjoyed learning about the histology of skin.